My name's Kendall Nettles, and I'm a professor at the Wertheim UF Scripps Institute for Biomedical Innovation and Technology. And I'm here today to tell you about my work on studying steroid hormone receptors, the estrogen receptor, androgen receptor, and how we're trying to develop new therapies to treat breast and prostate cancer and other cancers as well. So what I'm going to tell you today about is a little story about what's called structural biology. And this is a field that's at the intersection of biology, physics, and chemistry, which is why I'm here, because it's very relevant to what we're talking about. And this is a technique that lets you visualize individual atoms and molecules in a cell. And there are a variety of different techniques we can use. But this is really critical to having advanced our ability to design medicines and to understand really basic things about biology also. So, you know, we're really focusing on making these new therapies, but we're also learning things about, you know, nutrition and diet and other things that are also important. So, you know, in our current age, we're having an epidemic of, well, old age diseases, which are chronic diseases. And so this is our focus, is trying to use these new approaches that have come out in the last few years to try to improve human health. So why is this relevant to quantum computing? Because we're going to talk about the combinatorial problem, which is trying to study all the different things going on in a cell, and then how do we, now that we can do that, how do we move on to the optimization problem, which is trying to figure out what's driving different activities. So let me tell you, first of all, about a little bit about the ecosystem that I've been operating in here. I heard this about the quantum ecosystem in one of the earlier talks. So we came here in about 2004, 2005, and this was work that was done by former Governor Bush and also state legislatures, our local county commissioners and local politicians and a number of other business development groups to bring us here to develop an institute that's going to focus on curing these chronic diseases through technology, basically, and translational research. And so what happened is we came here, and it's been very successful. I'm going to tell you a little story about that. But about 3 years ago, 2002, we became part of the University of Florida. And so this was due to a generous gift from Dr. Herbert Wertheim, and this then enabled us to become the Herbert Wertheim UF Institute for biomedical innovation and technology. And it's really made a transformative difference in my career. And so I think this is important for what everyone here is interested in, is that this has really enabled a type of team science for us where we're able to interact with the Cancer Center. UF Health is a multi-billion-dollar hospital system, so we have access to clinical trials. And I should also mention that for the last 20 years, we've had one of the top 10 graduate programs in the United States in biology and in chemistry. And so this has been very important in that we're training the next generation of leaders here, also through internship programs with undergraduates and high school students to develop the next generation of people that are going to be able to have the right skills to treat the relevant diseases. So let me tell you a little bit more about that. One of the most important things that we've gained access to is the expertise in all of these different departments at the University of Florida. So it's the computer science, it's the engineering, the chemists, but also in the computer science aspect of this, the University of Florida has the largest supercomputer in academia. So this is a surprise to a lot of people. I talked to some of my colleagues at Cornell or University of Chicago, and they don't have access to some of these computing resources that we do here. So you may have heard of this little company called NVIDIA. Well, the founder went to the University of Florida, and that's why we have basically unparalleled access to these resources that other people don't. And And so we can just do these large-scale computer simulations that other people don't have access to. All right, so let me move on to the science part of this. So when I came here in 2004, 2005 from the University of Chicago, that was really the center of hormone research at the time. But this was the old school. It was one man doing, usually a man also, doing the research, and not the kind of team science that we're doing now. And so this is where they discovered that if you chemically block hormones, that it can help treat breast and prostate cancer, which is still the main treatment today. The person who did that, Charles Huggins, is my basically grandfather in academia. And I'm going to tell you a quick story before I move on to my science, just to wake you up a little bit. So I don't know if any of you know where hormone therapy comes from. So your testosterone replacement therapy, estrogen therapy, the idea for tamoxifen, blocking, in prostate cancer. So Charles Huggins was aware of this. In 1880, a guy named Berthold was interested in male reproductive development. And so what he knew was that if you removed the testicles from a young chicken, it would never develop male characteristics, the beard and the comb. Now, I don't know what inspired him to do this, but what he did next was to take those and put them in another young chicken. All right, it's a little weird, but that chicken then immediately grew male sexual characteristics. So you've learned one thing you can take home and tell, your kids or your family today, which is where hormone therapy comes from, which was surgery. That's how we used to do science. All right, so when I got here, we were moving on from doing things in a test tube, and they promised me that when I came here, they were going to have this ability to measure everything at once. And so this is kind of the theme of my talk, is this is the way we were doing it back then, was with robots. We're actually going to try to measure everything at once, and we're going to build a machine that can do 1 million test tubes in a day. By moving racks of these things, we make a little plastic deck that has 1,000 test tubes in it, and so on and so forth. And so we're going to solve the problems of science by basically engineering and measuring everything all at once. As it turns out, this becomes something that we can solve with AI, is to try to understand and integrate all that information. But what we can't do is see it all at the same time. And I'm going to go on to explain why that's important for drug discovery. So the technique that we were using 20 years ago now, don't forget, this is when the human genome was first sequenced. Oh, I have to teach you one thing other than about chickens, I'm sorry. Which is the basic dogma of biology, which is that your DNA is comprised of little sections called genes, and each gene makes a protein. And so why is this important? Because the proteins are most of the drug targets, and these are what's doing most of the jobs in the cell. So what's a protein? It's a series of chemicals that are strung together. So the estrogen receptor is a protein that has about 600 amino acids. Each amino acid's a little chemical grouping of maybe 20, 30 atoms. And it folds into this machine that can then, like any other machine, it takes energy and it does stuff. So what does it do? Well, this particular machine, it folds around a hormone, and then it turns on or off other genes. So it binds to your DNA, and it turns on the genes that make a breast and a uterus, or the testosterone genes will make the male reproductive characteristics. All right, so what we've done originally is to study this is called X-ray crystallography. So we want to take a picture of this machine, and so you've got we can make it, and it's in a liquid, and you take a picture, and what does it look like? Well, nothing. It's invisible, right? How do we make this so we can see it? Well, if you make it into a crystal just by dehydrating it, think about salt water. You can't take a picture of salt in solution, but if you make it into a crystal, you can shoot very small light waves, like X-rays, that are kind of the same size as the atoms, and they'll reflect off the atoms in a way that it's amplified by the crystal, and we can make an actual snapshot. Okay, so this sounds great. We can visualize all this, and we can feed all this into the AI of Google and build AlphaFold. So what's this? This is something that we've trained now with all the data, so we can do the combinatorial problem. We can measure what every protein looks like and feed it in, and then we gain the ability to predict new things that we hadn't seen before. But this is the problem, is that all it has is these snapshots from the crystal structures. So you're trying to get a picture, let's say, of a dancer doing a dance, and all you get is this. And that's it. And you're trying to infer all the rest of it from this. So what happens with AlphaFold? If you have a protein that has two different kind of shapes it takes, what you get is an average. So if you're doing this and this, what you get is this. It's not really that helpful in many ways. And so this is where the quantum aspect of this is going to come into play. And so we can now simulate on this supercomputer tens of thousands of atoms moving and dancing. And so the question is, why can't we just look at that and figure out how it works? And I'm going to give you an analogy to sports or dancing, but I like the sports one for this. So imagine swinging a bat, and you want to make an equation or a simple representation for how can you actually hit a baseball, for example. Or let me give you another analogy. This is an old, maybe a myth, but Bruce Lee apparently could put his hand on a board and move 1 inch and break a board. And so how does he do that? It's an energy transfer. And so when you think about how do we visualize this with X-ray crystallography, you just get a picture of the hand swinging the bat, and that's it. So can you tell who can hit the baseball just from looking at the hand, or do you need to maybe move back up with the arm a little bit? Well, any of you that have ever done sports or had children will remember this coach in your ear saying what? It's all in the hips, kid, right? It's a motion that starts in your foot, and it goes into your hips and into your hand, and it's a cooperative motion involving all of it all at once. And people have different ways to hit that are efficient. And so this is the problem with trying to understand how this thing is dancing. And so let me paint you, I'm going to finish off by painting you a picture of what this thing looks like as a molecular machine. So it has two kind of stalks or feet that stick to the DNA, and then it has a little body, and then just a head on top with a mouth. And I think of this thing like a Venus flytrap. So it eats the hormone, and it kind of chews on it for a while, and then that changes the shape of it. And then what that shape does is it lets other things come in that are the other proteins that actually decide whether to turn the gene on or off. So it's dancing, it's got different partners, and those partners are going to determine the outcome. So why is this so complicated? Well, when it eats it, there's a lot of details in there with the water that's coming in, and there's electrons on everything that are just swirling around. It's not just a lock and a key like you heard about in school where it's a solid thing, it goes into this hole, and that's how a drug binds. You have this moving object that's moving over time that has these electrons that are whirling around, and as the drug goes in, you've actually got transfer of energy through these electrons. And this is what we cannot currently visualize, is the different paths of those individual atoms and their electron structure, all right? And then the second thing is, as it dances in different ways, and we would like to know what are all the different ways that it can dance at one time. And then we can do what's called generative AI is the idea, right? You can design new things because you know how the whole system works. And so in order to do this, you need really a huge infrastructure. So we need to be able to measure the genes and make the drugs and kind of input that hard data, the ground truth data, but then we need the computer systems, we need the talent locally. So I'm teaching a class, an AI class, remotely now at University of Florida for these statistics and computational science AI researchers. And it's amazing, just in 2 years, the changes I'm seeing. They are already knowing how to code, they understand the underlying statistical principles, and the infrastructure that they've built here to do this is really the only reason that we're making this progress. And I think I'm going to close on that. Thank you.